This is the fourth lecture in the Build a Cell Chemical Reaction Network modeling series. Today I'm going to talk about stochasticity in chemical reaction networks. I'm William Poole, and this is actually a topic very related to my research. Just to recap what we've talked about in some of the past lectures, we've talked about deterministic chemical reaction networks. A chemical reaction network is a set of species. Here I'm calling them X, and reactions, which take a form sort of like this, where a set of input species given on the left-hand side is converted to a set of output species at some rate or propensity rho of X. And in deterministic dynamics, these are interpreted as differential equations, where the concentration of the species of Xi changes based upon summing over all the reactions. Now, in chemical reaction networks, you might imagine that there's actually stochasticity, and this stochasticity can come from many different sources. One of the sources is small molecule counts, which will give rise to the stochastic dynamics of a chemical reaction network. Partitioning between volumes, which we'll cover at the end of this lecture briefly, which might happen, say, during cell division, and also measurement noise, which we're not going to cover in this lecture, but is a very important source of experimental stochasticity. Stochasticity really matters in biology. I have three examples here. On the left, we see an example from an Elowitz paper where they measured the gene expression in single cells. And you can see the cells here growing and dividing and changing color as different genes are expressed. And they actually measured the production rate and got a histogram of values, a distribution of protein production rates. And this distribution is probably caused by variations in the molecular characteristics of each cell, how many plasmids it has, or how many ribosomes, or something like that. So the production rate actually is a random variable, not a constant single value when you look at each individual cell. Similarly, if you do experiments and look at the actual counts of molecules, so this paper by Long Kai measured the number of proteins, not fluorescence, but actually quantified the numbers of proteins, you see that proteins are produced in bursts, where maybe a single transcript is made. That transcript produces many different proteins before it's degraded, and then the next transcript is made. You can see that bursting behavior right here on the, you can see the bursting in this sort of like step function nature of this plot. And they can actually calculate the number of protein molecules made per burst, which is a distribution. And finally, stochasticity can actually affect deterministic behavior. So this is a really cool synthetic system of a whole bunch of DNA oscillators that might involve some enzymes as well. And what they found is that even though each oscillator has deterministic dynamics, the stochasticity of partitioning caused each droplet to have different dynamics from all the other droplets, even though they have the same molecules inside of them. So in other words, some droplets have more of one species than another, and that stochasticity led to different dynamical behavior at the deterministic level, showing that partitioning between volumes can also lead to really pronounced stochastic effects. The statistical fluctuations are the basis of stochasticity and CRN. So we're really thinking about molecular collisions here, where the time between collisions is a random variable. So if I have two molecules in a box, there's going to be some, and they're moving around, jiggling due to Brownian motion. Eventually, they're going to hit each other, and that's sort of the basis of a chemical reaction. Two molecules collide, and the reaction occurs. And if I have just two molecules, the amount of time it takes for them to collide really is going to depend upon a lot of things. It might depend upon where they're located right now, their velocities, the temperature. And you can really model this as a random variable, where on average, if you do this experiment where you watch two molecules collide over and over again, that's the expected time of collision is going to be constant. but there's going to be a lot of variance in that collision time, where sometimes it might happen very quickly in succession, sometimes there might be a long wait. And that means that this appears like a random number, and this gives rise to stochasticity when you have small counts. On the other hand, if I have a very large number of molecules in a box, and I wait for a collision to happen between two of them, well, it might appear like collisions are happening all the time because there's so many molecules, they're always bouncing into each other. I don't have to wait at all. And in particular, the variance in the collision time will go to 0 in the limit of n to infinity, or the deterministic limit where there are many, many molecules. So this is the true difference between stochastic dynamics of a chemical reaction network and deterministic dynamics. In the stochastic case, we have small numbers of molecules, and the expected time between reactions is a random variable. While in the deterministic case, reactions are occurring continuously, and there is no waiting between them and no randomness. So this gives rise to the stochastic dynamics of a chemical reaction network, and we'll consider an example network here. A goes to 2B and back. So we're going to convert concentrations that we use in the deterministic case to the probability of molecular counts, and continuous changes in concentration, which is our ordinary differential equation, become discrete stochastic changes in counts. So what does this mean more concretely? Well, if you think about concentrations, these are continuous variables. A concentration can be any value between 0 and that's greater than 0, really. So it's a continuous value. I can have a concentration of 1 or 1.2 or pi, whatever I want. It's a real number. While counts are discrete variables. I can't have half a molecule. I can only have 1 molecule or 2 molecules or 10 molecules or 1,000 molecules. So counts are integers, positive integers. And if you simulate a stochastic CRN in this way, you get sort of this behavior. Here you see the dotted lines are the deterministic concentrations, and the solid lines are many trajectories of the counts changing. So where the deterministic simulation has lots of intermediate values, the counts are always integer values, and they jump discretely between different integer values. So counts are on the integer lattice, and concentrations are real numbers. And again, this is showing you the deterministic simulations and the many trajectories, so just examples of the dynamics of a stochastic CRN. If I average over these stochastic dynamics, I get a probability distribution. And a probability of counts is a map from combinations of discrete variables to continuous variables. So what does that mean? That means I can have some probability that I have NA of molecule A and NB of molecule B, which is really a probability distribution defined on my entire integer lattice, which goes to a probability, which is a real number. So when you look at that, what does that look like? Well, it looks like a distribution like this. So here, for each point on the integer lattice, I have a probability of a certain number of A, and that changes with respect to time. And so these are really vectors where every point in time I have lots of probabilities for all of the different possible states I can be in that are varying with respect to time. And we can write down this as an equation called the chemical master equation. So again, we have this probability vector of giving some counts x, which is a big vector. There's lots of different numbers of molecules I could have, potentially infinite. And it's changing in time according to this equation, which looks fairly complicated, but all it's really saying is that I have outgoing probability, which is the probability that I'm in my particular state x times all of my reactions leaving that state. And I have incoming probability, which is the probability that I'm in a state that's one reaction away, and the probability of that reaction occurring and putting me into the state that I'm in now. So formally, this is called a Markov chain, if you're familiar with that stochastic model. And let's just go through a quick example. So again, if we look at this example, A goes to 2B at rate K1 and 2B goes to A at rate K2, we can write out the probability. It's going to be a derivative. And we have the outgoing probability, which is the probability that I'm in this state NA and B at time t. And I have K1 times NA, which is the probability that this reaction occurs, and K2 times NB times NB minus 1, which is the probability that this reaction occurs. And this looks a little bit different than the propensities we saw before, which might have been NB squared. This is called a stochastic mass action propensity, which is a slightly different form. And then I have my incoming probability. Again, there's two reactions. One of them is the probability that the first reaction occurs. So I start with 1 more Na and 2 less Nb, and that reaction occurs at the rate K1 times Na plus 1, and that will take me into my state Na and B. Or I could have 1 fewer Na and 2 more Nb, and the second reaction occurs at this rate, bringing me back into the state Na and B. So this gives me a big differential equation where my probability vector is changing with respect to time. And we can actually write that out very succinctly as a vector times a matrix. So the change in the probability vector is equal to some matrix Q times the probability vector. And this looks very, very nice. And you could even say, oh, this is a linear ordinary differential equation, which has a solution that looks like this. So you could write out the probability of a vector x at time t is equal to some initial condition times e to the q, where here we're using the matrix exponential function, which if you don't know about that, just look it up on Wikipedia. It's pretty straightforward, and a function of time. That looks great, but that's actually too good to be true. Typically, P and Q are way too large to handle and often even infinite. And so I don't really know how to deal with infinite matrices and vectors with a computer very well. So that makes this a very hard equation to deal with numerically. And to make that, give you an example, here's a very, very simple CRN that has infinite vectors. If I have nothing goes to A and A goes to nothing and nothing goes to B and B goes to nothing, here's my probability distribution. I have, a probability that I have 0 of both. I have 1 A and 0 B's, 2 A's and 0 B's, all the way down to an infinite number of A's and 0 B's, or I could do the same thing, 0 A's and 1 B, all the way down, or n A's and n B, or an infinite number of both. This is a very, very huge infinite vector, which its derivative is given by this infinite by infinite matrix times another infinite vector. And although this equation looks really nice, in practice, you can't really deal with vectors that are very large. And in fact, even for cases where it's not infinite, the size of these vectors grows exponentially with the number of species in your chemical reaction network. So if you have more than 2 or 3 species or 10 species, like many of the models we have, these vectors are way too big for you to handle quickly on your computer. Instead, what people do is they use something called the Gillespie stochastic simulation algorithm, or SSA. And this algorithm is going to exactly sample trajectories from stochastic CRNs. So instead of trying to solve that really complicated infinite dimensional equation, I'm going to say, how does my CRN behave? Let's get an example of what this behavior looks like. And these are similar to the trajectories I showed you in the second or third slide. And then we're going to use those trajectories to calculate statistics. So how does this algorithm work? I start at some state x of t. I'm going to have some amount of time I wait till my next collision or my next reaction occurs. And that's an exponentially distributed random variable tau with a time constant lambda, which is given by the sum over all my propensities. And this makes sense. If my propensities are faster, my reactions are happening at higher rates, I have to wait less time for the next reaction to occur. I now choose a reaction proportional to its propensity. If a reaction happens at a faster rate, it should be more likely to occur than a reaction that happens with a slower rate. And then I update my trajectory. X of t plus tau is equal to x of t plus the change of molecular counts as a discrete change due to the reaction that occurred. And that's it. I do this over and over again, and I get a trajectory. And so in the workshop, we'll be using bioscrape to simulate stochastic dynamics. And it uses exactly this algorithm. And importantly, once you do this, you get these, you probably do it many, many times to get lots of trajectories, which you might think of as different experiments or different data points. And you can then use these ensembles of trajectories to approximate distributions and calculate statistics or learn other things about your stochastic CRN. And we'll go through in the examples some of the nuances of how to actually deal with lots of trajectories. Now, one thing that people are often interested in when dealing with stochastic CRNs is the steady state distribution. And this is just when the probability distribution stops changing. So P of x dt is equal to 0, which you could also write out in this way. And informally, you might think of steady states as the stochastic equivalent to stationary points or fixed points or attractors that you have in a dynamical system. In the deterministic case, there's sort of an analog there. More formally, you can also think of the steady state as an eigenvector of this matrix e to the qt with eigenvalue of 1. And so this is a very formal way of thinking about it, but it's helpful for analysis. And if you don't know about eigenvectors and eigenvalues, that's okay. You don't need to use any of what I'm saying right now to actually understand stochastic CRNs. But if you do know about this stuff, I think this can be a helpful perspective. And what's really interesting about this is that the other eigenvalues and eigenvectors govern the relaxation of the CRN. So how long does it take to reach steady state? Well, that will be a function of your other eigenvalues and eigenvectors. And the steady state is the eigenvector with eigenvalue 1, so it never changes. And steady states are typically computed numerically via SSA. So you run a simulation for a very, very long time. And eventually, as you calculate statistics over that simulation, and you get a steady state distribution. So I've just talked to you about stochastic CRNs and given you a very fast overview of how to simulate them and how they work. The next part of this talk is on another source of stochasticity, which are fluctuations causing partitioning. And so what is molecular partitioning? Well, if I have a small number of molecules in a box, they might not appear evenly spaced. So recall that previously we talked about what is the sort of collision time, which gives rise to stochasticity. Well, the spatial distribution gives rise to partitioning. And here you could imagine cutting this box in half. And if these two molecules are bouncing around and moving, it seems pretty plausible that I could, if I did this experiment many times, see 3 different cases. I could see 1 molecule on each side, 2 molecules on one side, or 2 molecules on the other side. And on average, I would expect that the number of molecules on the left-hand side minus to equal the number of molecules on the right-hand side plus. But because all of these cases are possible, there is variance in this distribution. They're not always equal. In some cases, I'll see that there's 2 on one side or 2 on the other side. And that gives me variance. However, when I have a really large number of molecules and I start doing the same experiment, I won't see any variance. So I can cut this large number of molecules in a box in half over and over again. And again, I'll expect that the same number of molecules will be on each side on average. And importantly, in the limit as my number of molecules goes to infinity, the variance in the number of molecules that I count on either side will go to 0. So partitioning is another effect that occurs when you have small numbers of molecules. And it occurs a lot in biology because you have encapsulation. So you might imagine I have some large volume here with volume V and a number of molecules N in it, and it becomes 2 smaller volumes, maybe via cell division or some sort of vesicle formation or budding process. And the smaller volumes have a volume of V1 and V2 with an N1 and N2 number of molecules in them. the simplest way to model the stochasticity in this partitioning is using something called binomial partitioning. And this is just the binomial distribution, which is a very well-known distribution from statistics, where the probability that I get k molecules in my first partition up here, that n1 equals k, is given by this equation. And you have this factor p, which is v1 over the total volume, which is basically saying that, if v1 is smaller than v2, p is smaller than 1 minus p, and I have a larger probability of going into the larger volume. So this is just sort of saying that my probability should be proportional to my volumes, or my relative volumes. It's worth noting that this is sort of assuming that my molecules are homogeneously distributed in my volume. There's no spatial clustering or clumping of molecules. And in some models where you might know that, oh, molecules actually tend to aggregate with each other, or maybe there's something like a liquid-liquid phase transition, you might actually get non-binomial partitioning, but binomial partitioning is the right place to start. It's the simplest. And a really interesting sort of mathematical fact about molecular partitioning is that it's very difficult to differentiate from the intrinsic stochasticity in a CRN I talked about before. So if you actually work out some math, and there's a great paper here by Poole and Paulson, you can show that, given sort of typical experimental data, it's very hard to tell if the stochasticity you're going to observe is from partitioning or from stochasticity in a chemical reaction network. And in practice, what this means is maybe you have to look at both of these effects to really understand your system, especially when you're dealing with cells and small volumes with small molecular counts. So this sort of brings us to a very important point, which is, when do you want to use stochastic simulation? So when you have a small copy number of molecules, as a rule of thumb, n is less than 20. And this is actually surprisingly common in biology. If you are engineering a bacteria, you frequently have fewer than 20 plasmids. And if you have a genomically integrated gene, you certainly have, probably a single copy of that gene. If you're investigating something like noise and variance or stochastic effects, obviously you probably want to use stochastic simulation in your model. And the data you collect is really important. So if you're collecting single cell data, like from microscope videos or flow cytometry, you will have data that is amiable to stochastic analysis and can match your stochastic simulations. On the other hand, you probably want to use deterministic simulation if you have larger numbers of molecules. And really, over 100 is enough to say, well, if all of my molecular species are over 100, I'm probably not going to see a lot of stochastic effects. Again, we're not talking about all of our species, we're just talking about some molecules. So if one piece of DNA is at copy number 1, that's enough to give rise to large stochastic effects. But a bulk experiment, like a cell extract experiment or an in vitro experiment, unless you're doing it in a very small volume, is almost certainly deterministic. You also probably want to use deterministic simulation when you're investigating bulk or average behavior. So if you don't have data from small volumes from single cells or small vesicles, like you have plate reader data or you're looking at populations of cells in some way, you're not going to be able to see the fluctuations and noise in the individual cells. So there's really no reason to do stochastic simulation. And finally, deterministic simulation is much faster than stochastic simulation. So if analytic and numerical tractability are important, you probably want to focus on deterministic simulation. So let me give you a quick summary, then I'll take some questions, and then we'll move into the a sort of coding demo. So stochasticity is observed in vivo and in vitro in biochemical systems. And stochastic chemical reaction networks model fluctuations in molecular counts and collision times, while deterministic chemical reaction networks are assuming everything is continuous and there is no noise. Stochastic chemical reaction networks are formally Markov chains, which represent the probability distribution of the species counts. and is given by this equation here. Unfortunately, this equation is usually infinite dimensional, or at least very high dimensional, so you can't solve it analytically or numerically, and instead you use the Gillespie SSA algorithm to approximate the probability distribution and its steady state by taking many trajectories and averaging them. Another source of stochasticity is molecular partitioning between volumes due to fluctuations in spatial distributions. And partitioning is hard to differentiate from the fluctuations in collision times. These things look very similar mathematically. And in our workshop, we'll be using bioscrape to model both chemical stochasticity and partitioning. So here are some references. There's a lot of great literature, experiments, and theory out there, which I highly recommend you look at if you're interested in this. Yeah, so there are definitely hybrid simulation algorithms out there. Bioscrape that we're using does not implement one. They're typically approximations, so you have to be a little bit more careful about when they're accurate or not. But there are many, many different, there's also a type of simulation called Langevin simulation or stochastic differential equations, which is going to simulate a concentration with noise added to it, like Gaussian noise, which is another way of doing a hybrid simulation. The challenge is that deterministic simulation is really assuming there's an infinite number of molecules, and stochastic simulation is assuming there's finite numbers of molecules. So if you have reactions that involve both those species, it can be hard to reconcile. If I have an infinite molecule and I use two of them, how does that change my finite number of molecules? So it can be a little bit tricky to do. And I've personally found that in most cases, you can use one or the other to good effect. And I would like to acknowledge all of the co-instructors, the developers who've helped on the software, the TAs, and the Build-A-Cell PIs, and the NSF who's supporting this.